The overall goal of this procedure is to isolate extracellular matrix from cultured cells for a wide variety of downstream experiments to help understand functions and properties of extracellular matrix in normal tissues and disease processes.
This method can help answer key questions in the extracellular matrix field, such as the organization, molecular composition, protein interactions, or functional properties of extracellular matrix. Also, the extracellular matrix can be prepared from any adherent cell type.
The main advantage of this technique is that it can be carried out at different scales to efficiently remove cells and retain extracellular matrix proteins.
Demonstrating the procedure will be Silvia Rosini, a graduate student in my laboratory, and Andrew Hellewell, a postdoctoral researcher.
To begin, plate cells on cover slips for the imaging of live cells and extracellular matrix, or ECM, for fluorescence microscopy studies of fixed ECM, or for preparation of cell-derived ECM for small-scale functional studies. Incubate the cells at an appropriate temperature and humidity for a suitable time for the cells to deposit ECM, typically greater than 16 hours. In an extractor hood, prepare 20 millimolar ammonium hydroxide in a suitable vessel by using deionized water to dilute the stock solution 1:14. Remove the cells from the incubator and gently remove the culture medium. Then add PBS without calcium or magnesium by gently pouring against the wall of the dish. Rock the dish twice and pour away the liquid. Then repeat the wash 2 more times. In the extractor hood, remove the